how can IgG4 antibodies, the antibodies that we see show up in majority of the mRNA vaccinated individuals, potentially be contributing to cancer? That's the topic of today's video. We started a series related to this topic of how IgG4s might be contributing to cancers, including concept that is referred by colloquially as turbo cancers. That term doesn't exist in medicine. In medicine, the closest maybe example of that would be what we can refer to as hyperprogressive disease. And in today's video, we'll talk about, in more general terms, how IgG4 could potentially be contributing to cancers in general, as well as we will elaborate on the topic of the turbo cancers and hyperprogressive disease, basically. My name is Dr. Mikola Rashek of Neurogenomics. Let's get started on this important topic. So, as I mentioned, hyperprogressive disease or turbo cancers is a very new concept in medicine and oncology. It has only been discovered within the last decade. The first description of it in scientific literature is only in 2017 and has came about from use of medication against cancers. Basically, the use of medications, they are referred to as immune checkpoint inhibitors, okay? And while such treatment can help certain individuals, it was discovered that in subgroup of those who are treated, it can lead to very rapid cancer growth. Now, this has obviously sent like shockwaves in the medical community as to what is happening. It led to confusion as well. There was basically not full understanding what might be happening, what is the actual true cause or phenomenon behind it. And merely in the last 5 years, do we have scientists, clinical scientists, providing us certain mechanisms that might explain this. So in the previous video that dedicated to this topic, I talked about the very first paper I came across that gave any suggestion. That was only in 2019. Today we'll talk about a paper that came out in 2020. So just 4 years ago, by a group from China that provided additional very important, valuable information. Basically, the authors of that paper mentioned, we really don't know how IgG4 still could be contributing to cancer, and they wanted to basically investigate this further. And this is how we're now gaining this very important knowledge, very valuable knowledge, but still, it shows you how little do we actually know with regards to IgG4s, how IgG4s could potentially be dangerous, in what way could they be dangerous. And clearly, we have a big gap of knowledge here that we need to massively expand on. And I have a feeling that in the coming years, we will gain a lot of knowledge on account that we are unfortunately seeing this unexpected side effect post mRNA vaccination, where so far with only minor exceptions, we're seeing these unusual IgG4 antibodies show up in all of the individuals who took the mRNA vaccines for COVID-19. All right, so then let's talk about this particular paper. So what did the authors do? They did both. Like in previous paper I discussed, these guys also looked at both patient data as well as they did certain experiments with mice. This is very common. That's a standard in science where you want to basically enhance our understanding of molecular biology by working with animal models. So let's talk about the patient data first. What did these guys do? They took about 82 patients who had an esophagus cancer, and they compared them against healthy individuals, and they measured the antibody levels in the blood. And what they measured is basically, what they were able to show is that those who had cancer had higher levels of IgG4 in comparison to healthy individuals. And here's a key number that this is extremely important, because remember, we have such little amount of information on this topic. the healthy values were approximately 3% of IgG4 in blood, versus those who had cancer was about 11%. Why is this important? Because this is the first value that at least gives us some sort of a metric as to perhaps what should we be looking out for with regards to measuring IgG4 levels in those who might want to worry about this issue, and where should there maybe alarm bells go off. So that's very important. And more importantly and more crucially, what these authors also were able to show is that these IgG4 levels increased the further the staging of cancer increased. So cancer, when cancer is being diagnosed, you can diagnose it based on how not aggressive, but how late in its development is. And the later it is in its development, you can label it with a certain stage. It goes from, say, for example, stage 1, 2, 3, and 4. And the later the stage, basically the more dangerous the cancer becomes in general. And what these authors were showing, that the later the stage, the more IgG4 antibodies were being seen in these patients. Now, going back, speaking of which, they also mentioned a little bit of background on IgG4s, and really there's so little information when it comes to cancers. And they did mention that there was one article in the past of authors who suggested that IgG4 ratio between IgG4 and a total amount of IgG antibody in patients' blood was prognostic of recurrence of cancer, so basically cancer coming back. So, and then there was another group of authors that they mentioned who showed that IgG4 that target specific cancer elements could compete with IgG1 that also target elements, and this is how potentially IgG4 could be contributing to cancer development. That's pretty much the only thing they really provided as a background. So a lot of this information is very new knowledge. This is emerging field, as you can see, and there's literally just few papers that I can discuss in these video series on this topic. But what these authors also did, they also looked at actual tumor samples of these patients, and you can look under microscope, and you can stain the antibodies, so that basically you can have a certain chemicals attached to the antibody, so that it allows you to see where the antibodies target by chemical reaction that basically changes the color, and where the stain normally would look bluish hue or pinkish hue, if you see these brown or dark reddish, that's where you can detect where the antibodies are binding. And what they did, they looked for basically how many lymphocytes were present in patient tumors. Lymphocytes, basically immune cells. In this case, they were looking at B cells that produce IgG4. By the way, B cells that produce antibodies, they're no longer called B cells technically, they're called plasma cells. I always refer to them as B cells for sake of simplicity. Basically, you can trigger B cells to eventually produce antibodies, and they have a different name. I always refer to them as B cells just to keep it simple. They looked basically for B cells that were producing would be producing IgG4. The tumors were infiltrated in cancer individuals. However, they looked at the area around, there was no infiltration. Around the tumor, there was no infiltration. And same thing with IgG4. They also looked for IgG4 inside the tumor itself in comparison to the areas around tumor. And basically, there was 4 times increase of IgG4 in the areas around the tumor in these patients in comparison to next to the tumor or anywhere else, biopsy anywhere outside the tumor. Okay, so clearly showing that IgG4s might be playing a certain role, because there is an elevation of these IgG4 antibodies in that tumor environment. but it's the next experiment that is especially interesting and really key. So what they did is they took 22 patients of different cancer types this time. So they looked at patients with esophagus cancer, breast cancer, as well as colon cancer. They isolated both IgG1 antibodies as well as IgG4 antibodies from these patients. And then they took the actual biopsy of those patients' cancers, and they looked how these antibodies that these isolated from the very same patients, how do those antibodies react with those cancers? And they were clearly able to show that IgG1 was binding to it. They could see that color reaction, the color chemical reaction. They attach a specific chemical to these antibodies called biotin. Biotin is this molecule that is chemical that is super reactive, and you can use it for molecular biology because you can attach other things to it, and that's how you can then produce the color, which allows you to basically see what you want to see in a tissue sample. And they were able to show that way that the IgG1 antibodies isolated from the cancer patients were targeting the actual cancer itself of the very same patients, which is great. That's what you want. Why do you want that? Because basically when you have antibodies binding to a cell, it targets that cell or those cells for destruction. That's how we clean up cancers in our own body whenever we accidentally might produce cancers, which is not infrequent. Why? How does it happen? It's because once the antibody attaches itself, say, to a cancer cell or a pathogen in that matter, like say a virus, it's the stalks of the antibody that remain exposed, and it's the stalks that are grabbed by other immune cells, and then you can have destruction of the either cell or the pathogen. So remember, let's just briefly recall this, because this will be important for this paper as well. Antibody is like in a letter Y, right? The tips of the letter Y, these tips right there, they will bind to whatever the antibody is supposed to be targeting. That's called antigen. So those tips is called antigen binding fragment, and abbreviated as FAB. And the stalk below that, the basically flat part, is called FC, a region FC component. C stands for either component or crystallizable. I've seen both terms used in literature. And it's the FC component of antibodies bound to IgG1 antibodies bound to cancers that will now be recognized by immune cells and destroy cancer. There's at least 3 different ways of how pathogens or cancer cells can be destroyed. It's called antibody-dependent cellular cytotoxicity. So natural killer cells can recognize these, grab these, and will destroy the target. There's also a process called antibody-dependent cellular phagocytosis. Phagocytosis means other immune cells such as monocytes or macrophages can come in, and they will literally swallow up the entire complex and destroy it inside. And there is also a concept we refer to as antibody-dependent complement deposition or activation. And basically, complement is a system floating in our blood that allows to build up on top of these antibodies, build on top of it like pieces of Lego, which then marks it for destruction as well. And this will become important in a moment. So IgG1s of these cancer patients were binding to their own cancers. Great. IgG4s did not. This is important because because this basically meant that it doesn't matter what IgG4 targets. What's dangerous if that IgG4 enters the tumor environment, because the same IgG4s that can end up in the tumor environment and are indicative of tumor growth, they don't even actually target. In any way, they don't attach themselves to cancer cells, which means basically it doesn't matter what these IgG4s targeted. And as you will see in a moment, it's their stalk that matters. And it's the next experiment that they also did. What the authors showed is that how these IgG4s could be dangerous potentially is because the stalk Fc component of IgG4s can bind itself to the stalks of IgG1 antibodies. Now, we've known this for a long time. This has been discovered literally decades ago, but it's never really been understood in the context of cancer development. And in this case, the authors were able to show that they could bind IgG1 of these cancer patients to their own cancers, and then they were able to detect IgG4s on top of the IgG1s. So, and that meant they had to be binding via those Fc components. So then, and what they also were able to show is that they could take the Fc component of the antibody, IgG4 antibody, and that was enough to bind to the IgG1 as well. This way they were able to show that IgG4 can interact with any IgG antibody, IgG1, IgG2, IgG3, and IgG4. Basically via that Fc component, via the stalk. So the stalk of IgG4 can bind to a stalk of any other IgG antibody, and when that happens, it will remove that antibody from basically participating in its normal biological activity. In this case, it would be, for example, protecting against cancer. It will, however, IgG4 will not bind to the stalks of other types of antibodies such as IgG A, M, or E, or D. And it's also independent of the species. So they were also able to show that they can show the same interaction between IgG4 stalk and IgG antibodies between species as well. So very, very important experiment. Next one is also very important because they were able, what they did is now they took blood from patients, and they wanted to see whether IgG4 interferes in any way with interaction of immune cells by grabbing to IgG1. Because if IgG4 via its stalks binds to other antibodies such as IgG1 stalk to stalk, will it block then these important biological events that are supposed to clean up the cancer? So they took the immune cells from the blood of patients, and they were able to show that once they added IgG1, IgG1s were able to interact with the immune cells of these patients, such as monocytes or macrophages. But that interaction could be prevented by addition of IgG4, exactly like they were predicting. And it was in a dose-dependent manner. So that means the more IgG4 they added, the less interaction they were able to observe. And basically, addition of IgG4 prevented those biological events that are supposed to be clearing out cancer, such as that cytotoxicity. So it reduced the cytotoxicity, measured cytotoxicity against cancer cells when IgG4 was added to IgG1, and it also prevented that complement deposition as well. So basically, it prevents biological function of how you're supposed to be fighting cancers. And again, they were able to show that the Fc component of IgG4 was enough to achieve that. So that's the key here. So you don't even need the whole antibody, IgG4 antibody. As long as if you have that letter Y, you cut the tips off, the FAB component, antigen binding component of the antibody, the Y part, cut it off, it's gone, and you only have the stalk remaining, the Fc component. That Fc component of IgG4 antibody is enough to start interfering with proper function. So very important experiments showing how IgG4s can interfere with proper biological functions that are supposed to be protecting us from cancer development. Now, we're gonna move on to the next series of experiments that they did with mice. However, I gotta tell you, this is a trigger warning because this type of information that I'm describing, well, all these animals have to be sacrificed. Luckily, the experiments doesn't last long. It's just a matter of days, but it can sound gruesome. So just forewarning, okay? What they did in these experiments, they took mice and they injected cancer cells underneath their skin, and basically when you inject cancer cells, eventually tumor develops, and then basically you induce cancer development in these mice by injecting these cancer cells. So what they did is they injected mice, normal mice. They were just like a regular mice. There was nothing unusual genetically about them, and they injected breast cancer cells in these mice, and they were able to show that that resulted in tumor development, as cancer development as expected. But, and this is key, when they co-injected the cancer cells, breast cancer cells, with IgG4, there was an increase in tumor development, increase in size. So, and this was IgG4 that would not even target cancer cells. So that IgG4 was just something completely different. Again, it does not have to target cancer cells to have this effect. And they were able to show that it did this. And when they isolated IgG antibodies, so any IgG antibody except IgG4, they did not see that effect. So the tumor size was just like as if they didn't add anything. So IgG4 specifically was increasing the tumor size. And it's very visually explicit. You can see how the tumors that they were then cutting out of these mice of course were euthanized, and they were able to cut out the tumors and measure the size, etc. of the tumor, and you could see that IgG4 influence the tumor size. They repeated this series of experiments a couple more times in different ways as well to show how these IgG4s could be involved in cancer development in these animals. So this time, in the second round, they injected different cancer cells in mice, and I believe it was colon cancer cells, but they did additional experiments. What they did is they injected these mice with either it's just a liquid so that's your control, that's a sham, or they injected IgG4, and again you saw the increase in tumor size, or they injected IgG1. Surprisingly, they also saw tumor size increase, and they also injected just the Fc component. By the way, the IgG4 they were injecting in this time around was the same IgG4 antibody that is used in that cancer treatment in real patients, in real life. It's called something like nivolumab, and it's one of those treatments for immune checkpoint inhibitor treatment. So they used the very same one, and they also split it in half, they cut it in half, and they were able to show once again, so they just simply used the Fc component, and they were able to show that when they just added the Fc component, the development of tumors, of cancer in these mice was the biggest. So again, it's the biggest impact. So they were able to show this, basically in terms of in different ways. And in previous paper I was talking about on this topic, remember, the authors were testing the FAB component, showing that if you cut the antibody in half, FAB component, meaning the tips of the antibody, that didn't have effect. These guys did the reverse. They showed that it's the Fc component that is inducing this. So, and then the last set of experiments that they did, and again, visually very obvious, very explicit, how these cancers are growing in these mice because of the IgG4 antibodies. And the last experiment, they used skin cancer model, so it was papilloma cancer. And in this case, they weren't injecting anything in the tumor site. I believe they were just injecting into the bloodstream. So they used IgG4 to inject directly into the bloodstream, and that increased once again increased the number of tumors showing up in the skin and tumor sizes as well, and beyond what control would be. So basically it increased made it all worse. And they also did infusion of IG antibodies in general. So they did what's the word I'm trying to look for? IV, basically, infusion of these antibodies straight to the bloodstream, and that actually reduced the tumor sizes. So it's protective in this case, without IgG4 present in that injection. And so they used 3 different mouse models showing how IgG4s could be leading to the development of cancer. So, and the reason why obviously they focus on that immune checkpoint inhibitors is because of the fact that they wanted to see, could the FC component of that anti-cancer treatment be responsible itself for progression, the hyperprogression that is seen in some of the patients as well. And clearly they're suggesting that is what is responsible. So now we have 2 different groups. These guys were mentioning, listen, we're the first ones in the world to show it's the FC component as well. It was suggested by the group from Italy previously. They were the previous year, which was the information I discussed in previous video, but these guys from China showed for the first time that indeed it's the FC component. So they're describing, look, now we have new molecular understanding of what might be happening in these cancers and how IgG4s overall could be contributing to cancer development. So totally new mechanisms now we have, and this they confirmed that indeed 2 things could be happening. Indeed, you could be blocking certain immune cells, protecting cancer patients. You can block their protection with these IgG4 antibodies. And and I forgot to mention, they also tested for binding for the FC component receptors. So those FC components, when they interact with different immune cells, there's different types of receptors, and IgG1, they were able to show binds to whole bunch of different receptors. They looked at FC receptors version 1, 2, and 3, and IgG1 was able to bind all of them. IgG4 was only binding one type of them, but IgG4 is preventing binding to all of those receptors by IgG1. So they basically reconfirming the theory from 2019 that IgG4s can block immune cells from performing their protective functions, and as well as, this is the new information, IgG4s can not only just block receptors on these immune cells, but can also block antibodies on cancer cells themselves that are supposed to target cancer cells for destruction. They can bind to those antibodies via FC component, via those stalks, and block those antibodies from being able to call in the army to start protecting you from the cancer growing in size. So 2 new mechanisms. We're going to expand on that further. So like I said, there's only a few papers published on this topic, so not much literature. So in the paper, in the next video, we're going to expand this further as well. What else can we know about this topic? Because this is so important in the context of now seeing increasing levels of IgG4 antibodies targeting spike protein in the mRNA vaccinated individuals, in case this might be a threat. We don't know this. This might not be an issue. And I can tell you that I participated, I went to a medical webinar on IgG4 related disease, basically people who produce IgG4, too much IgG4 in their body. And it's a disease you can treat. There's a treatment for it. And I can tell you that those experts proclaim that they don't think this is an issue, this increase of IgG4 antibodies against spike protein post-vaccine, they actually stated, and I was absolutely shocked, they stated they think it's actually a good thing. But we need to find out still what does it mean, because as I said, this paper clearly showed these authors, they tested everything. They tested antibodies, IgG4 antibodies that they could buy from different stores. They tested IgG4 antibodies that are used for anti-cancer treatments and whatever they, or IgG4 antibodies isolated from patients. No matter what they used, when they co-injected these IgG4 antibodies with the cancer cells in mice, it increased tumor size. We still don't know whether that could be replicated in humans. Remember, mice and humans are not the same thing. Sometimes, yes, we use animal models to learn a lot, but what you observe in animals does not mean will be observed in humans at all. So it clearly points to the fact that we need to be studying this in great detail. And I'm pretty sure, because of the increasing interest amongst the scientific community to start studying these IgG4 antibodies observed post-mRNA vaccination, I'm pretty sure we will be learning more about their potential contribution or their lack of in cancer development. But because this is such new knowledge in terms of how this might be happening mechanistically on a molecular level, I wanted to basically start providing you this information in great detail. All right, that's all I have today. Well, wasn't short. None of these videos are short because it's so complicated to explain everything. But thanks for your attention on this topic, and thanks for hanging out with me on this beautiful day in nature. And please share this video. This is how we grow. Please subscribe to the channel if you haven't already, so you can be notified when I provide additional videos on this important topic when it's in terms of IgG4 contribution to cancer development. And please leave a comment. Let us know what you think. And finally, also please check out our Patreon account as well, where we post additional topics that do not make it to YouTube. All right, bye everyone. Ciao for now.